Good morning, good afternoon, and good evening. I'm Tom McNulty, and I'm here with our presenter, Angela Criswell. Thank you all for attending Rigaku's webinar series, X-ray Computed Tomography for Materials in Life Science. This is our fifth segment, Plant Applications. It will focus on using the principles and techniques covered in past webinars and their application to plant science. Before we start, a few housekeeping items. Please note, if you've missed any of our previous webinars, you can view them on the Rigaku website. When today's presentation ends, you'll automatically be directed to a Rigaku webpage containing the links for the previous webinars. Now, as far as today goes, we will usually be taking questions during the live webcast. We ask that you submit your questions via the Q&A button on your screen. We will not be monitoring the raised hand or chat features. We will be saving your questions submitted during the webcast, and we'll answer as many as we can during the Q&A session at the end of the presentation. If for whatever reason you have difficulty viewing the webinar live, please note it is being recorded and you will be able to view the recording beginning tomorrow. Okay, that said, I would like to introduce our speaker for today, Angela Criswell. Welcome, Angela.
Thanks, Tom, and thanks for everyone for joining us for the webinar series, X-ray Computed Tomography for Materials in Life Science. And today we will talk about plant science applications. In particular, we'll use concepts learned from previous webinars and apply them to imaging plant materials. So as you probably remember, if you've attended previous webinars, I referred to X-ray imaging as a form of photography, and it is. But if we decide that we want to X-ray image or photograph something, first we have to decide what we want an image of. So in this case, let's consider that our subject is a dandelion, and the dandelion has a diverse life cycle, so we have to decide what part or parts of the life cycle we want to image for the dandelion. And for those life cycles, we could, for example, image the flowers. We could examine a seed head. We could also image the delicate seeds themselves. Regardless of the parts we choose, we will find that our experiment can be compromised because these samples can and will move during our experiment. So maybe we need to consider some extra hardware or steps to minimize movement caused by external effects like air movement. And secondly, it's very likely that you're not interested in a single image of the dandelion, but are instead interested in how the dandelion changes during its life cycle, or how it changes in response to some external stimuli. Plant researchers often want to understand how plant morphology changes, so in fact they may perform many experiments across a number of experimental variables. So the key is that one image is often not enough. For this webinar, we'll review considerations when performing CT experiments with plants. And in particular, we'll talk about sample movement and size considerations. We'll talk about instrument optimization. And then finally, we'll look at some applications of X-ray imaging with respect to plants. So as we've already mentioned, sample movement can be a difficult challenge during X-ray imaging of plants. Sample movement has a number of causes, all of which trace back to the fact that we are looking at living organisms, and these organisms often have fragile, flexible, or even elastic parts. So plants are living, growing organisms, and as a consequence, they're constantly moving. And we also know if we take a cutting from a plant, the plant will lose some turgor pressure or stiffness, and it will start to dehydrate or wilt. So we'll have to consider this problem when imaging cuttings. So what can we do? Well, first, we can consider shorter data collection times. And then second, we can look at ways to secure the plant or plant part that we want to image to minimize that movement. Thirdly, we may need to have a setup that leaves as much of the sample intact as possible to maintain turgor pressure and sample hydration. So let's look at each of these options in more detail. First, let's look at an example of the impact of data collection time and we'll consider this for a delicate sample. In this case, we might expect that there's an increased likelihood of sample movement as data collection times get longer, so our hypothesis might be to collect images as quickly as possible to mitigate movement. So let's look at that. On the left is an image slice from a CT data set. This is a 3D model of a flower. The full data collection time in this case was 57 minutes, and we can clearly see artifacts caused by sample movement. For example, there is some doubling of petals and on the leaves. These effects are even more pronounced if we looked at a cross-section image. So here again, we see the doubling of the petals and the leaves. So can we rescue this experiment by using shorter exposures? So here's a data set collected for 14 minutes. In this case, we still see the doubling like in a longer data set, but our cross-section image shows less movement occurred. Instead, in this case, the movement manifests more like blurring. So there's still sample movement on the scale of a 14-minute experiment. What about even shorter data collection times? So in this case, the total data collection time on the right is 4 minutes. And in this case, we can resolve a single flower. A cross-section of the petals and the stamen also appear much clearer. And this suggests that we could collect a quality data set for this sample just by collecting at a shorter exposure time. So one thing you'll probably notice with the image on the right is that there is more noise for this short image compared to the long image. So your ability to use short exposure times will really depend upon the instrument and instrument settings that you use. So what you can do before you start any longer experiment is do a test for and evaluate the degree of movement for your sample. So in that case, you would collect multiple still projections with, say, 30-second or 1-minute intervals. And after some length of time, you would subtract image 2 from image 1, and image 3 from image 2, and so on. And if there's movement, there will be signal observed in a subtracted image. When the subtracted image has no signal, this means that the sample is no longer moving relative to your earlier image, and that would be your cue that you can now start data collection time for your longer data collection. So it turns out this works great for some samples, especially for materials, which are usually hard samples, but it doesn't always work for plants. So in that case, what other options might we have? So your first or next consideration would be to evaluate how your sample's mounted during the imaging experiment. So this slide shows a number of different tools we use in our imaging lab to secure samples. So for example, the mung seed on the left is secured to the sample mount using dental wax. This works great for some samples, but any sample with a rounded or non-flat mounting surface area may move over time, even with the use of wax. So in that case, we may want to utilize paraffin, like shown in the middle. So in this case, paraffin was used to stretch around the sample, around this mung seed, and then wrapped further around the pin. And it turns out paraffin works great in many cases, and conveniently, it's very X-ray transparent. Another common mounting tool we use is UV adhesive. So on the right-hand side, a small piece of carbon fiber is secured to the sample mount using a UV cured adhesive. We just apply a small amount to the sample or the mount and then use UV light to cure. And most of these will harden in just a few seconds and form a quite tight bond. In some cases, the shape of the sample may require that we have some sort of holder mechanism to secure the sample. So in those cases, we can often just have a look around the lab to find useful tools. In our imaging laboratory, it's not uncommon for us to utilize common lab items like microfuge tubes, pipette tips, and other plastic containers. We also use foams, falcon tubes, plastic bottles, and other items. So if we just take a look at the things around us in the lab, we can find some useful items to hold samples for our experiments. When using any new material, a good first experiment is to collect data on the container or adhesive material alone, or with a small plant sample to evaluate its X-ray transparency. Keep in mind that our plant samples contain light elements like carbon and nitrogen and oxygen. However, some adhesives or materials could contain heavier elements. So the goal of this preliminary experiment is to identify X-ray transparent sample holders. And in some cases, you may be able to create your own impromptu sealed sample environment. So on the left, I show a Microfuge Tube used to hold our mung seed in a way to keep it from drying out. So first, a small amount of water is added to the tip of the Eppendorf tube, and the water is held in place by surface tension, so then you can invert it, place the mung seed inside with a small amount of soil to minimize movement. And then the lid of the sealed Eppendorf tube has a nice flat surface so that we can easily secure it on our X-ray imaging mount. In some cases, you may not be able to create the contained environment for your sample, or you may want to image the whole plant. So there are a number of instruments that will accommodate whole plant imaging, so it's definitely a doable experiment. And in that case, we take advantage of the non-destructive nature of CT imaging, which can be quite powerful in that we don't need to remove the plant or roots from soil. We don't need to take a cutting of the plant, and we don't need to cut open fruit to analyze seeds or fruit material. Nevertheless, the image on the right does bring up an important consideration, which is sample size. So imaging the full plant requires that we have a suitable instrument. So let's consider sample size, as this will impact what instrument we collect data with, as well as the data collection settings we're going to use. In some cases, the allowable field of view may be smaller than the plant height. And in that case, we can move the sample or collect at multiple heights. So experiments such as these may require that we register and perform CT image stitching to reconstruct the whole plant. So I'll briefly address that case. The next important thing in this case is that sample size will have a big impact upon our CT experiment and instrument needs. So let's take a look at some candidate plant samples, starting with very large plants or trees and work our way down in size. So in the first webinar that was given by Aya Takase, she presented a very early Rigaku instrument that was used for imaging trees. And in this case, experimenters at the University of Tokyo Institute of Industrial Science were able to non-destructively image trees and determine the age of the tree. But the prevalence of full tree imaging isn't a really common application. Instead, plant researchers are looking to optimize specific morphologies, usually during the growth and development cycles. So in that case, full-sized, smaller plants may be the imaging candidates. In these cases, the length scales can still be in the millimeter to meter range, but they're significantly less cumbersome than trying to image a large tree. So as you can see in the example here, a tomato plant was imaged, and it shows its root system was isolated for further analysis. In this case, a researcher could be interested to understand root branching. They could be interested in understanding soil penetration, nutrient flow, or some other characteristics. Also, the root body itself may be an important food source, and thus an important candidate for study. In some cases, researchers may be interested in the fruit of a plant. So for example, one might want to understand fruit abundance or fruit size, maybe branching like in the case of grapes, or maybe the nutrient content may be of interest. And in this example, I show a corn husk. Corn and maize are significant crops and humans deeply depend on them. So we'll look more at corn later in the webinar, but you get the point. fruit bodies can be important candidates for CT imaging. Well, what about smaller things? How about seeds? Well, there are lots of interesting morphological characteristics of seeds. And we can look at single seed morphology, or we can compare seeds among different plant variants. We can also look at populations of many seeds at one time and do comparative studies. We may also want to understand how a seed changes during germination or how it changes with storage age. So seeds are very interesting candidates to researchers. Regardless of the sample you use, a good start is to understand the size of the samples you want to image, as well as the feature sizes you ultimately want to see in your plant sample. So the next step will then be to consider, for my plant sample of size X and feature size Y, what instrument will I use? And of course, a good first question will be, which instrument? Well, let's take a look at two candidate samples. And on the left, we have our corn sample again. And in this case, the features range in size from sub-millimeter to several millimeters, in this case, 5 millimeters. And our field of view, in this case, is a total of around 32 millimeters. And if we compare that to the right, where we have a seed, the feature sizes are in the range of a few microns to tens of microns. And our total field of view in this case is around 2 millimeters. So it would make sense to think that you may choose different instruments or beam geometries for each of these samples. So, for example, the corn sample was collected using cone beam geometry, and the seed was imaged using parallel beam geometry. Often, facilities may have several instruments that may be better for looking at some samples compared to others. So it's always a great idea to consult your local X-ray expert if you're unsure about what instrument to use for your sample. And if you want to learn more about the beam geometries that I've mentioned, please refer back to one of our earlier webinars for more information. So now you have an instrument selected, you next want to consider other questions. How will I mount my sample? Will I need to take a cutting of my sample? Will the full sample or plant fit into the instrument? And shown on the right is our tomato plant, and shows it fits well in this particular instrument. Also, we need to consider what X-ray energy will I use, and what field of view will I use. And importantly, the questions to ask are, what feature sizes do I expect to see in my CT images? And the feature sizes will influence your choice of voxel size used for the experiment, because you'll want to use a voxel size smaller than at least half or less of the size of your smallest feature. That way you can significantly capture feature details in your final CT image to allow you to carefully characterize and analyze that sample. So to learn more about voxel size and how it impacts CT image file size, feature size quantification, and other considerations, have a look at previous webinars on the Rigaku website or YouTube. So as we mentioned earlier, you may also need to collect at different lengths to image a full plant or the plant parts if the whole plant is larger than your field of view. And in cases like that, it may be necessary to register and then stitch CT images together to get a full picture. And this slide shows a quick example for okra in which two parts of the okra were imaged with a smallish field of view. In this case, the sample is positioned to have some overlap of the okra sample in each scan, and this overlap is represented by the blue box you see here. Because these regions are shared in each scan, we can move CT images after reconstruction to identify a suitable overlap to calculate the proper registration of the okra. And the key consideration in this case, where you must stitch images together, is to make sure there are unique features in each scan that allow you to register samples. If no unique features exist, as in the case, let's say, for a material with a repeating pattern, you might want to consider adding a second material to the side, maybe tape it on to act as a fiducial, and we've done that in our laboratory. So this slide shows the okra after registration of the two scans and stitching the images together. And the cross-section image shown on the right shows the inside of the okra and the seeds. And so notice that here the stitching is successful in that we have intact seeds and the edges of the okra surface are smooth and without a break. So it turns out that the features inside plants make registration and stitching less difficult than, say, with some other material samples. So we've looked at a few considerations related to imaging plants and plant materials. What about some real samples? Let's look at a few examples. And let's start by revisiting the tomato plant we showed earlier. So in this case, a full tomato plant was imaged. And as you can see, the instrument on the right shows that it fits perfectly fine in the instrument. And in this particular experiment, tomato plants were grown in two different soil types. And the question in this case was which growth media was best for our CT imaging experiments of these plants. So the tomato plants were imaged with identical settings on the same instrument. On the left side, the tomato plant was grown in a basic peat mix, while on the right, the plant was grown in coconut coir, which is an organic growing medium that's supposed to be really good for holding water well. So these 3D images from the CT experiments show that it's much easier to isolate the root system in the peat mix sample compared to the coir mix. In fact, it's possible to use simple window leveling to isolate the root on the left side here, where window leveling does not isolate the root from the coir sample on the right. So this experiment identifies the peat mix as a better growth media for CT imaging and makes life much easier for later root segmentation. And it turns out there are wide choices of growth media for plants, but this experiment, the simple experiment, illustrates an example where soil is a consideration and you can explore it and exploit it via using CT methods. Now let's revisit our corn husk sample. In this case, the field of view is small, so we translated the sample along the husk and collected overlapping scans to illustrate this stitching that I talked about earlier. So after collecting all the scans, the sections were easily registered by the fact that we have lots of unique features throughout the corn material, and that allowed us to register and stitch together computationally this corn husk. Now we can manipulate it and analyze it in full. So what types of questions might we want to learn about corn? Well, first, we may want to look at kernels, and to do that, we first have to isolate each kernel so that we can compare one kernel to another, or different populations of kernels to one another. So in this case, I used a watershed transform to identify each kernel, and then we can start to do further quantitative analysis. And this slide shows a simple example where we look at the Z position of a kernel. And things that are closer to the bottom of the husk are red and have lower Z value. And we use a rainbow lookup table. And as the Z value increases and we get away from the bottom of the corn, the kernels turn blue or purple. So as you would expect, kernels at the other end are purple or blue. But kernel position may not be so interesting. Perhaps other characteristics would be more interesting to quantify. So how about kernel size or kernel volume? This slide shows a kernel size distribution plot on the left, and this histogram plot depicts the size frequency of kernels versus the volume. And on the right, you see an overlay of the matching lookup table on the 3D model of the full husk and with a cross-section. So we could ask questions about where is kernel size largest or smallest? For example, we can look at distributions of kernels. We might compare this to other types of kernel husks or different corn varieties. So you can see that we can ask a number of questions about the sample once we have a full image to evaluate. We could also ask questions about the corn silk because we can see it quite clearly, or we might ask questions about the corn husk itself. So lots of things to explore here. Now let's look at some CT imaging examples for seeds. So this slide shows a couple of examples of seeds. On the left are mung and millet seeds. Mung seeds are the green seeds and millet are the smaller yellow ones. Not all seeds are small, so for an example, I show you on the right some sea beans, which are commonly also referred to as drift seeds, and these are much larger. So we'll come back to these seeds in a second, but first let's take a look at some small ones. And recall that the millet seeds are the small yellow ones shown in the red box. It turns out that millet seeds have quite complex characteristics. So for example, this shows a cross-section slice from a CT image, and it clearly shows the hull, germ, endosperm, and other structures of the millet grain. And for those of you unfamiliar with millet, these are small-grained, warm weather seeds belonging to the grass family. They're annuals, and they are highly drought tolerant, and they have a nutrient content that's similar to cereals. So as you might expect, they're an important crop for investigation, and many groups throughout the world perform research of these important samples. And this slide shows a millet seed with an apparent crack. In this case, we may want to characterize these cracks. To do so, we can segment out the crack as depicted on the right, and then carry out some calculations on the crack. We may also want to expose seeds to different environments and analyze them for the presence or absence of cracks. We may want to expose them to different temperatures or store them for different lengths of time, and again do this analysis. So there are lots of things we can look at. And let's look at a different type of question. Perhaps we want to understand the characteristics of the seed hull or coating, as this may have some impact on its tolerance in certain environments. And this slide shows the segmentation of the seed hull from which we can calculate or look at the hull thickness or volume. And in fact, in this case, our hull volume is 1.5%. So there are a number of things we can quantify when looking at these millet seeds. And now let's go back and look at our larger seed example. So for those of you unfamiliar with sea beans, these are a species of, these come from a species of flowering plant native to Central America, the Caribbean, South America, and Africa. And these plants are notable for having some of the world's largest seed pods, and they can measure 12 centimeters across and 2 meters tall, as shown by this gentleman holding one of these seed pods. Inside the seed pod are anywhere from, say, 10 to 15 seeds, and each of these seeds are also quite large and can have a diameter of about 6 centimeters and a thickness of about 2 centimeters. Well, let's have a closer look at these seeds. I picked up a few samples from a local beach area and collected some CT images, and all of them look pretty much the same on the outside or in. Some are polished, some are not. And if you look at a CT image, they look pretty consistent. You can see that they're pretty empty inside. They have this large air cavity, and so they may look fairly uninteresting on first glance. And after reading about these, I turned out that these air cavities are absolutely crucial for the plant's survival. So the seed's hollow cavity gives the seed buoyancy, and after the seed pods fall from the tree and the seeds are freed, they are washed by rain into rivers and ultimately into the ocean. And these seeds can drift really long distances on ocean currents, making their way to beaches of other countries, nearby countries. So as you can see, the seed hull is quite thick in this case. And that means that with the buoyancy and these incredible resilient seed pods, they can last a long time. So as a matter of fact, they can float for about 2 years. and land on a faraway shore and start to grow. So these are truly remarkable plants that have evolved to withstand some really challenging conditions and allow their wide dispersal across oceans and spread their genetic material. And because they spread so much, that's often why they're called drift seeds. And I'm pretty sure that all of you have likely eaten or held a really common drift seed. That's because a coconut is also a drift seed or type of seed bean. So next, let's look at a different type of seed. This is a cranberry seed, and these data were actually collected by my colleague Aya, and they're one of my favorites. They're pretty simple, but I really like all of the surface and internal complexity for the features that we see. And it's easy to see that the characteristics are more similar to the millet seeds we observed earlier versus the sea beans themselves. From an X-ray microscopy point of view, they can be quite easy to segment for further analysis. So we can look at some seed characteristics. So for example, the hull for this sample is quite complex compared to the millet seed. And we can calculate the hull volume, and it turns out to be 3.3%, almost a little over double that for the millet seed. Remember, it was 1.5%. And it's likely all these little protuberations add to the volume that we see for the cranberry seed to make for larger volumes. We also see voids, like in the case of the sea bean, but they're less at about 9.9%. So finally, let's look at some CT data for a leaf. And on the right side is shown this small leaf sample, and on the bottom is a quick radiograph taken for this leaf. And it shows darker nodules where there's greater X-ray absorption. So if we look at the tomogram, we see these as lighter green modules in the image. And specifically, one of the things we can identify is that these nodules are not on the surface, but instead they're spread throughout the leaf itself. When we look at an image that isolates these as regions of interest, for example, they're clearly throughout the leaf, and we can start to ask questions like, how big are these? So we can take an easy measurement with a ruler to look at the size of each of these. So here's the front of the seed, and this is the length of the seed. We see this, the leaf, and the leaf is about 154 microns thick. And if we look at each of the nodules, they're on the order of, say, 20 microns to about 50 microns in size. So we can easily take a measure. But if we want to compare these nodules amongst those on the same leaf or other leaves, we'll have to do something more sophisticated. So for an example, we'll have to isolate these, and then we can look at, say, volume histograms. Remember, this is a histogram plot of the frequency versus the volume. And this was done using the Dragonfly program, as were the other analyses. So some do appear quite small, while others are larger. And the same lookup table applied here in the histogram plot is applied over here. So this also allows us to pick up perhaps some errors that we see. So this large red bar corresponds to this nodule. And perhaps it's more than one nodule, and we may have to work a little bit more in Dragonfly to get these fully separated. So it's an easy way to visually assess your analysis that you're doing. Nevertheless, you can see the distribution of these quite well, and you can also see the veins quite well. So we see lots of material and things that we can look at further in this leaf sample. So with that, I just want to let you know that all of these images were collected on Rigaku instruments. If you want to learn more about these instruments used in the presentations, please go to rigaku.com and to the contact field to find a sales specialist in your area. And if you're interested in watching the previous webinars I referred to in this presentation, you can easily find them on our site by entering Rigaku webinar in the search field of the Rigaku homepage. And also, I want to make you aware of our next webinar on X-ray computed tomography, where Aya Takase will cover geology applications. That webinar will take place on July 15th, a Wednesday, at the same time this one was, 11 a.m. Pacific or 2 p.m. Eastern. Thank you.
Okay, Angela, thanks. Very, very interesting. And thank you so much for presenting. We do have a number of questions that have come in today, which we can answer one by one. So first one from Scott. Scott's curious to know what the material, what the root sample material was that it was growing in, because it seemed to be very X-ray transparent. That was from one of your earlier slides.
It was a simple peat mix from the pottery place where I got it from. So you'll see there were some specks in there. So they sprinkle a little bit of dirt as well as some organic material and even styrofoam. And it's got a lot of air, so it makes it nice and X-ray transparent.
I was going to answer it's a Home Depot material.
That's right.
But,
that's true, Tom. That's exactly. Just
Okay.
a local gardening store by my home.
Yeah. Okay, we have a couple of questions now from Ula. She's asked three in sequence. So first question was, which instrument was used to scan the millet seed?
The millet seed, in that case, we used both the Nano 3DX as well as the HX130. The high resolution images that you see in that case were from the Nano 3DX, and we used copper radiation in that case, 8 keV.
Okay. Next one from Ula was, were the cranberry seeds used dry?
Yes, they were used dry.
And also, was the leaf fresh? How long did the scan last?
So in that case, that leaf, I took some scans when it was fresh, and then it turned out it survived really well over time. So I ended up taking a number of images. So each of the individual images are on the order of 1 to 2 hours. And the nice thing is it was really stiff, didn't really move a lot during the experiment, so it made a nice candidate. And I tried to look at some herbs and things like that, but as you might imagine, as soon as they were cut, they started to wilt. But this was just from a bush right outside of our office here in the Woodlands. Okay,
interesting. David asked, which instrument was used to collect the cranberry seed data, and what were the source, and settings for the data collection?
Yeah, so in that case, we used the HX130, which is the same instrument where you saw the plant imaged in full. In that case, the energy setting was anywhere from 60 to 90 kV. I usually take a few scans to see which one's going to give me the ideal contrast, and then I'll do a longer scan with that setting.
Okay. Thomas asks, first a comment, says, hi, and thanks for the nice presentation. And she asked, would you be able to tell us about the estimated resolution ranges for the three instruments shown on a previous slide?
Yes, I can do that. So in terms of the minimum resolvable features for a Nano 3DX, this is going to offer you the highest resolution. So in that case, you can get to sub-micron, so 1/4 to 1/2 micron resolution. For the other two instruments, and by the way, that one uses parallel beam geometry. The other two instruments that were shown use cone beam geometry, and depending on the field of view, the resolution can change to a few microns to many microns in that case. Sometimes, in some cases for the experiment shown, the voxel size was on the order of 50 to 100 microns.
Mm-hmm. Okay, next one from David. Setting includes voxel size and data collection time. So actually, I think he was looking for answer to his question previously. What were the voxel size and data collection time?
Oh, they varied for each of the samples. If you're interested in a particular one, by all means, please just drop me an email and I'm happy to fill you in on the X-ray energies that I used and the voxel size, data collection time, for any of these. I'm happy to share that as well as the data sets themselves.
Okay. Next one is a comment from Monica, and she is understanding what the examples. She understands that the samples shown have been hydrated, and she says in her group they have followed the protocol for preparing samples for SEM, which is dehydration and drying.
Yes.
Just comment on that. She's asking if that's correct.
Yes, that is correct. And with CT imaging, we're fortunate enough that we don't really have to do a lot of sample prep like you'll have to do in SEM. The disadvantage is that some cases we are contrast challenged, but there are numerous examples of different contrast enhancing agents that you can use. So for a matter of fact, I don't know if Keith is here from the Danforth Center, but he has some literature out there that I've read where they've used iodine, and there's a number of different examples. So by all means, our sample prep is quite easy compared to yours.
Right, okay. Another similar question again from David says, I'm using contrast agents on seeds, and I find that iodinated compounds work at about a 30 milligram per gram tissue concentration level. I would appreciate any advice.
I honestly don't have much for you. I have very limited experience with them. I tried a few things that I thought I would introduce in this webinar and just didn't have time to complete them. But I would be really interested in chatting with you about that further. Perhaps we need a webinar on just talking about contrasting agents.
A good future topic.
Yes, definitely.
Ula has another comment question. How do you create contrast between peat and tomato roots? And also thanks for answering our previous questions.
Yeah, I think in this case, I got lucky, to be honest. And so one thing I will say is, a lot of these seeds I got from the Sprout people. So I'll just give them a little shout there. So basically you can order sprouts, you hydrate them, and as soon as they start to germinate, just eat them. And they recommended that if you wanted to grow larger plants, that you use this coconut coir sample. Well, it turns out, like I mentioned, that is really good for holding water. And because it holds water and it's, a very similar organic makeup to our roots, that's why we can't see anything. So, the general fact that there's so much air, some styrofoam, and really light things going on, I'm going to guess as a way of the gardener to reduce their cost by reducing the amount of soil in those tomato plants, I got lucky. So. Okay,
very honest answer.
Yeah, hopefully it answers your question. So I by all means, I am not a plant specialist. I'm a specialist in X-rays. And, I was just reaching out for samples around me or some that were noted in the literature quite often to discuss as a part of this webinar.
Okay, Jerome asks that he wants to make sure that you were using Dragonfly for data processing, which I'm guessing you were.
Absolutely.
He's asking, how does this work, for plant images? Is there what software do you recommend for plants, if not Dragonfly?
I use Dragonfly exclusively to look at each of these. It was quite powerful.
Okay.
So for example, the watershed transforms were done with that. I will say for a couple of these, especially that corn plant was, I had to use deep learning to segment those kernels. They are all touching and they have a large surface area that touches. So they were a bit tricky, but they were a slam dunk if you had a great teaching data set with deep learning.
Okay. Jerome has another question. He's asking if there's a fix, I guess software-wise, if you cannot avoid movement in the plant samples. Are there workarounds for that?
Well, I think you're always going to be challenged. I mean, the problem is that as the sample moves, you'll start to see some streaking behavior. And then you really have to ask yourself, is what I see a true characteristic of the plant, or is it just a factor of the moving? And at some point in time, you may have to fix it into some solid material. So I've read some papers where people have embedded things in epoxy or plastic when they absolutely can't keep them from moving. But that example I show you for the flower, it was a rose petal, and it was really delicate, and it was really powerful that we could collect data as quick as 4 minutes. And as a matter of fact, that particular instrument is capable of collecting scans in as little as 5 seconds. I certainly didn't try it. I was just looking at different exposure times to get to the point where I would not see any of the blurring or shadowing that I saw. But yeah, it's really difficult. Plants are alive, and by virtue of that, they are going to move at some time interval.
Right, just like small children.
Correct.
So Keith has a comment, says, hi, Angela, and he's letting you know
Hi,
that
Keith.
they use iodine, I guess
Great.
he use Lugol solution, or phosphoric acid, or
Okay.
I'm sorry, phosphotungstic acid. And he says they are common contrast agents. So thanks, Keith.
Yeah, and by the way, I did purchase some Lugol solution based on what I read in from one of your papers, and trying it. I'm wondering if I need to back soak or those types of things. So like I said, it would be great to have a webinar topic just dedicated to that.
Okay, that's what we have for the questions so far. Anybody else want to send something in? This is the most questions that we've ever had for one of these webinars. So I really appreciate all the comments from everyone. Okay, well, I think with that, let's see what's next on the agenda. Yes, so with that, okay, that's, I guess, all we'll have time for now. So we will do our best to get back to all shortly if there are, any follow-ups to the questions that came in. I might have missed a couple as well. And as I said earlier, a recording of the webinar will be available tomorrow. An email will go out to all registrants with instructions how to view the recorded presentation. Also note that after the close of the webinar today, you will be automatically directed to a landing page on the Rigaku website with a link to the next webcast, geology applications. So I'd like to thank everybody again for joining us today, and we hope to see you next time. Bye now.